Hello students, I am continuing with chemistry of nucleic acids. Today I shall commence with RNA. We are discussing about another nucleic acid that is called ribonucleic acid or RNA. Ribonucleic acid or RNA is a polymer of ribonucleotides linked by 3' 5' phosphodiester bonds. Although its structure is analogous to DNA, but it has certain differences. RNA is usually single stranded. However, in case the complementary base pairs approximate each other, it folds upon itself, acquiring a hairpin like double stranded structure. But DNA has a double stranded helical structure. The second point of difference is that in RNA, the sugar moiety is ribose. RNA is susceptible to alkali hydrolysis due to the presence of 2' hydroxyl group. The sugar is in DNA, that is 2' deoxyribose, that does not undergo alkali hydrolysis due to the absence of 2' hydroxyl group. Third point of difference, that RNA contains the same purines as DNA, adenine and guanine. It differs in the pyrimidine components, that is, it contains uracil, and DNA contains purines, adenine, guanine, and pyrimidines. So cytosine is common for both RNA and DNA. So RNA contains uracil and DNA contains the pyrimidine thymine. RNA is relatively small molecule. There is 100 to 5,000 base pairs, whereas DNA is huge, containing millions of base pairs. It is essential that the total length of DNA occurring in the it is estimated that the total length of DNA occurring in the human body, that contains about 10 to the power 13 cells, and the length is about 2 into 10 to the power 11 kilometer, an astronomical number. And the fifth point of difference is most of the RNA occurs in cytoplasm in association with ribosomes and endoplasmic reticulum, whereas except a tiny portion occurring in mitochondria, it is largely confined to the nucleus. And next point of difference is, since RNA is a single stranded complementary to one of the two strands of DNA, the adenine content need not be equal to uracil and guanine to cytosine, thus it does not obey the Chargaff's rule. However, certain double stranded RNA, that is some viral RNA, are defined double stranded, these molecules obey the Chargaff's rule. But DNA obeys the Chargaff's rules. The double stranded DNA contains equal quantities of adenine to thymine and guanine to cytosine. RNA plays a vital role in protein biosynthesis, whereas DNA is the genetic repository. And finally, certain RNA molecules possess catalytic activity. They are called ribozymes, but DNA is non-enzymatic. However, recent research provides evidence in support of DNA acting as an enzyme occasionally. Now come to the different types of RNA. The messenger RNA, mRNA, transfer RNA, tRNA, ribosomal RNA or rRNA, heterogeneous nuclear RNA, hnRNA, and small nuclear RNAs, snRNAs. Now we are discussing one by one. Messenger RNA, that mRNA, it is synthesized as the primary transcript from the template strand of DNA and later processed to mRNA. They carry the information in the gene to the protein synthesizing system on ribosomes where the genetic information is translated into an amino acid sequence that is polymerized into a protein. The 5' terminal of mRNA possesses a 7-methyl guanosine triphosphate moiety linked to the rest of the molecule through an unusual 5' to 5' phosphate linkage. And this is known as the 5' capping. And this capping protects the molecule from the attack of 5' exonucleases. The cap is also involved in the recognition of mRNA by the protein synthesizing machinery. The protein synthesis also starts from the 5' end. The mRNA also contain methylated nucleotides apart from regular ones. Their significance is unknown. The 3' terminal contains adenylate residues that are about 22 to 50 and referred to as a poly-A tail, whose function is largely unknown. Perhaps it protects mRNA from 3' exonuclease attack. Now come to the transfer RNA or tRNA. Now transfer RNA or tRNA, transfer RNA is the adapter molecule that translates the information carried by mRNA into specific sequence of amino acids. There are at least 20 species of tRNA molecules corresponding to the 20 amino acids in each cell. A transfer RNA molecule contains 75 to 95 nucleotides. Its structure was first elucidated by Hawley. The transfer RNA molecule shows extensive internal base pairing and acquire a clover leaf like structure. And they contain a significant proportion of unusual bases, include dihydrouracil, pseudouridine, hypoxanthine. Moreover, many bases are methylated. Now, our tRNA contains mainly 4 arms. There are also another arm, variable arm. So these arms are, one is acceptor arm, is at the 3' end, you can see, and this area contains 7 base pairs, and the end structure is CCA, 3' end, that is CCA. 3' end of hydroxyl group, this 3' end hydroxyl group is forming an ester bond with the carboxyl end of amino acid. Now another opposite arm, this anticodon arm. So this is the anticodon arm of the tRNA. At the opposite side of the acceptor arm, it recognizes the triplet nucleotide codon. So that codon, the mRNA, the specificity of tRNA resides in the anticodon. The specificity of tRNA resides in this anticodon site, which has base sequences that complementary to that of mRNA codon. So that is complementary to this codon, GC, CG, CG, like that. The tRNA molecule will show specificity in both aspects, recognizing the mRNA codon as well as in accepting the specific amino acid coded by that codon. In this way, tRNA molecule plays a pivotal role in translation. The tRNA act as adapter molecules between mRNA and the amino acids coded by it. The nucleotides of codons have no affinity for amino acids. So the tRNA molecule act as mediators between the mRNA and the amino acids. Now the DHU arm or the D arm of tRNA, the D arm is shown because of the presence of the dihydrouridine. The dihydrouridine base, that area, DHU arm serves as the recognition site for the enzyme which adds the amino acids. Now come to the pseudouridine arm, but T size C arm. This is called this T size C arm. Here, the opposite arm of this D arm is the pseudouridine arm, as it contains called pseudouridine. Or psi, it generally denoted the Greek alphabet psi for pseudouridine. It is involved in the binding of tRNA to ribosomes. About 75% of tRNA molecule possesses short extra arm, that is called variable arm. Okay. The variable arm, you see the variable arm, based on this variability, tRNA grouped into two classes. Class 1 tRNAs is constitute about 75% of total tRNAs and contain 3 to 5 base pairs. And class 2 tRNAs, they have a length of about 13 to 21 base pairs. Now come to the ribosomal RNA, rRNA. These ribosomes are supramolecular nucleoprotein complexes. Several ribosomes comes together to form the polyribosome or polysome, providing platform for the protein biosynthesis. On the ribosomes, the mRNA interacts with tRNA, translating into the specific protein. The mammalian ribosomes have a sedimentation coefficient of 80s, S means Vedberg units. They are composed of two subunits, 60s and 40s. So 60s nucleoprotein units contain 5s rRNA, 5.8s rRNA, and 28s rRNA units. There occur about 50 different proteins also. And 40s nucleoprotein subunits contain a single 18s rRNA and about 30 proteins. So the S values are not exactly the additive when subunits are combined, because the sedimentation rates are influenced by the shape as well as the mass. The exact function of rRNA in ribosomes is poorly understood. Probably it plays a role in the formation of polysomes and in the binding of mRNA to ribosomes. Other types of RNA we have mentioned hnRNA, heterogeneous nuclear RNA, and that is actually the primary transcript of the mRNA synthesized from the template strand of DNA, and that is heterogeneous nuclear RNA. This is very large in size. hnRNA undergoes post-transcriptional modification in the nucleus and is processed as mature mRNA, which then enters the cytoplasm to serve as template for protein synthesis. The genetic information from template strand of DNA is coded into the complementary sequence of nucleotides of the mRNA, which is decoded in the cytoplasm by tRNA and translated into specific sequence of amino acids. The small nuclear RNAs, the snRNAs, are involved in the processing of hnRNAs into mRNA and regulation of gene expression. Another identical molecule, the small nucleolar RNA, SNO-RNA, is involved in the processing of RNA molecules. There are also small cytoplasmic RNA, scRNA, plays a role in directing the newly synthesized proteins to their destined locations. Now come to the RNA as enzyme. Yes, all enzymes are, we know the enzymes are proteins in nature, but recent experimental data suggests enzymatic function of certain RNA molecules. They are called ribozymes. Ribozymes, they are involved usually in the processing of newly synthesized RNA molecules to mature RNA. For example, you see the ribonuclease P. Ribonuclease P is a ribozyme involved in the post-transcriptional modification of the primary transcript of tRNA to mature tRNA. So ribonuclease P is involved in the post-transcriptional modification of primary transcript of tRNA to mature tRNA. Another ribozyme mentioned that peptidyl transferase, that is a component of RNA that plays a crucial catalytic role in the peptide bond formation. So peptidyl transferase plays catalytic role in peptide bond formation during protein biosynthesis. It is interesting to note that during early years of evolution, RNA acted as the early enzyme before proteins took up the role of biocatalysts. The metabolism of nucleotides, nucleic acids. As nucleic acids are made up of nucleotides, we describe metabolism of nucleic acid in the form of metabolism of nucleotides. Now, purine and pyrimidine nucleotides, they are present in the diet. They are not incorporated directly into the tissue nucleic acids. Humans synthesize purine and pyrimidine nucleotides directly from the amphibolic intermediates as per the body requirements. The metabolism means there is anabolism and catabolism, so synthesis and degradation. So first we will discuss about this metabolism of purine nucleotides. So first the synthesis or biosynthesis of purine nucleotides. The biosynthesis of purine nucleotides occur in two ways. One is de novo synthesis or de novo pathways, another is salvage pathways. De novo synthesis. De novo synthesis means de novo is a new synthesis of nucleotides occurs using the simpler metabolic precursors such as amino acids, ribose phosphate, carbon dioxide, and ammonia. And salvage pathways recycle the free bases and the nucleosides released from the nucleic acid breakdown. Now, de novo synthesis of purines. Different compounds contribute to the skeleton of the purine ring. You see the purine ring, this N1 of purine arises from the amino acid, amino aspartate, that is contributed N1 contributed by the aspartate. The second position is the carbon C2, and is the C2, and that is contributed to the N10 formyl tetrahydrofolate. Okay, and C8 by N5 N10 methylene tetrahydrofolate. This nitrogen atoms and third position and ninth position, that is contributed by this glutamine amide nitrogen or glutamine amino acids. Okay. C4, C5 and N7. So C4, C5 N7, and these atoms are contributed by the glycine amino acid, and finally that C6 originated from this respiratory carbon dioxide or bicarbonate or respiratory carbon dioxide. So these are the different contribution of the purine skeleton by different atoms amino acids. Now purine nucleotides are synthesized by many tissues in the body. The liver is the major contributor. The brain and RBC cannot synthesize purines by de novo synthesis. It should be noted that purines are not produced as free bases. Rather, they are formed as ribonucleotides, and the purines are synthesized piece by piece upon a pre-existing ribose phosphate. The pathway of reactions involves, and this pathway is described below. One is first the ribose 5-phosphate. This is the alpha D ribose 5-phosphate. That is, this is generated in the HMP shunt pathway of carbohydrate metabolism, and that serves as the starting material, the alpha D ribose 5-phosphate for purine nucleotide biosynthesis. It reacts with ATP to form the 5-phosphoribosyl alpha pyrophosphate, they call PRPP. So that PRPP is also involved in the purine salvage pathway, also in the biosynthesis of pyrimidine nucleotides. It is also used or utilized in the synthesis of NAD+, nicotinamide adenine dinucleotide, and NADP+, that is nicotinamide adenine dinucleotide phosphates. Also in the synthesis of histidine and tryptophan amino acids. Now second step is donates. Glutamine donates its amide nitrogen to PRPP, displacing its pyrophosphate moiety. Displacing the pyrophosphate moiety. The reaction involves the inversion of alpha configuration of C1 or PRPP, forming this beta 5. So from alpha it becomes beta 5 phosphoribosylamine, and thus producing the anomeric form of the future nucleotide. The NC bond formed in this reaction will eventually form this N-glycosidic bond linkage of purine nucleotide. The enzyme catalyzing the reaction is PRPP glutamyl amidotransferase, and that is the regulatory enzyme, and that is regulated by the feedback inhibition. So this PRPP glutamyl transferase is the committed step in this purine de novo biosynthesis, PRPP glutamate amidotransferase, and that is regulated by the feedback inhibition. So these two steps are very important. Next, this is phosphoribosylamine condenses with the glycine in the presence of ATP and to form glycinamide ribosyl 5-phosphate, glycinamide ribosyl 5-phosphate or glycinamide ribotide, we call it glycinamide ribotide. And this reaction, glycine provides the atoms which will become C4, C5 and N7 of purine ring, which I already have seen. N10 formyl tetrahydrofolate donates the formyl group to the free alpha amino group of glycinamide ribotide to form the formyl glycinamide ribotide, and the glutamine transfers the second amide nitrogen to the growing ring to form the formyl glycinamide ribotide. This reaction requires ATP. Okay. The purine imidazole ring is closed in an ATP dependent reaction to form this 5 amino imidazole ribotide. That is now the amino imidazole ribotide. The ring closure. Insertion of carbon dioxide. Insertion of carbon dioxide as C6 of purine yields carboxy alpha amino imidazole ribotide. Unlike most of other carboxylation reactions, this carbon dioxide does not require either biotin, this carbon dioxide does not require either biotin or ATP. Normally, the carboxylation reaction requires biotin or ATP. Here, this is not required. Anon of purine is contributed by the condensation of aspartate. You see, this next state is aspartate to this carboxy amino imidazole ribotide to form the 5 amino imidazole 4 succinyl carboxamide ribotide. ATP is required for this reaction. ATP is required for this reaction. The enzyme adenylosuccinate lyase clips this amino imidazole succinyl carboxamide ribotide to form this fumarate. So, and leaving behind the amino group of aspartate in the purine ring. The resultant compound is 5 amino imidazole 4 carboxamide ribotide. Reaction 8 and 9, they are identical to reactions in the urea cycle, in which citrulline is aminated to form arginine. So these are similar to this, identical to reaction urea cycle in which citrulline is aminated to arginine. The final atom of purine ring is obtained from this N10 formyl tetrahydrofolate from this amino imidazole carboxamide ribotide. Okay. Reaction 4 and 10, reaction 4 and reaction 10, this is blocked by reaction 4 and 10, and this is blocked by the sulfonamide drugs which inhibit the formation of this the tetrahydrofolate. So that tetrahydrofolate and these 4 and 10, they are blocked the sulfonamide drugs which inhibit the formation of the tetrahydrofolate. And the final reaction, you can say, of purine biosynthetic pathway is catalyzed by this cyclohydrolase, leads to the ring closure with elimination of the water molecule. Okay. The end product of obtained pathway is the first purine nucleotide, that is the inosine monophosphate. So this is the first purine nucleotide that is formed in the inosine monophosphate. From this inosine monophosphate, other purine nucleotides are synthesized. Yeah. And if you cannot remember, you remember this first 4 steps and then the final step. The 4, 5, 1, 2 is most important. They are more important. First 2, third, fourth, and then final step. If you cannot remember this, first 4, most of is 1 and 2, and then final step. So inosine monophosphate is the first purine nucleotide, and from which other nucleotides are synthesized. So this step you have to remember. This is the inosine monophosphate. Synthesis of this AMP and GMP from this IMP. The IMP formed in the de novo synthesis is rapidly converted to this AMP and GMP. Condensation of aspartate to IMP forms the adenyl succinate. Okay. The adenyl succinate is, this reaction is powered by the hydrolysis of GTP. So the energy that comes from the GTP. Now the amino group of aspartate is linked to IMP, and the remaining carbon skeleton is clipped off. As fumarate, resulting in formation of the adenosine monophosphate or AMP. The reaction is adenyl succinate lyase or adenyl succinase. Now IMP undergo here this step, then this is that adenyl succinate synthesis. Another is, you see this IMP undergoes dehydrogenation through reduction of NAD to form. Now NAD IMP dehydrogenase, it forms this xanthosine monophosphate. Similar to reaction 5, will get glutamine transfers the amide nitrogen to the xanthosine monophosphate to form this guanosine monophosphate, and utilizing this ATP. Resultant product is GMP, guanosine monophosphate. So for the synthesis of AMP, the GTP is required. For the synthesis of GMP, ATP is required. Now formation of purine nucleoside diphosphate and triphosphate. Nucleoside monophosphate. So the nucleoside monophosphates AMP and GMP have to be converted to corresponding. So AMP and GMP, the nucleoside monophosphates, will be converted to the corresponding di and diphosphate and triphosphate, and to participate in nucleic acid synthesis and other metabolism. And this is achieved by this phosphoryl group transfer from ATP. So this ATP donates the phosphate group by a relatively base specific this NMP kinase, and the NMP kinase catalyzes this reaction and forms the nucleoside diphosphate ADP and GDP, and then NDP, nucleoside diphosphate kinase, and again the ATP, and that catalyzes this reaction to form this ATP and GTP, nucleoside triphosphate. It should be noted that the various energy releasing reactions drive these kinase reactions further. ADP is converted to ATP primarily. This ADP is converted to ATP by oxidative phosphorylation and also by reactions of glycolysis cyclic cycle by this substrate level phosphorylation. So GTP serves as a precursor for the synthesis of tetrahydrobiopterin, which is required as a cofactor in the hydroxylation reactions of aromatic amino acids. The GTP that serves as a precursor for synthesis of tetrahydrobiopterin is required as a cofactor in the hydroxylation reaction of aromatic amino acids like phenylalanine, tyrosine, tryptophan, and also in the formation of nitric oxide. Yeah. Now come to inhibitors. We have mentioned already sulfonamides. We have mentioned inhibitors of the purine synthesis. Now we have seen the vitamin folic acid plays the crucial role. We have seen, we have already discussed the 4 and 10, that is that tetrahydrofolate and mention. We have seen this 4 and 10 and actually and 10 formyl tetrahydrofolate. So the folic acid plays a crucial role in purine biosynthesis. You have seen number 4 and number 10 reaction. Sulfonamides, they are structurally similar to PABA, that is para-amino benzoic acid, and PABA is a component of folic acid. So sulfur drugs, these sulfur drugs are used to inhibit the synthesis of folic acid by the bacteria, and this affects the synthesis of purines. Definitely purine synthesis is impaired, and nucleic acids finally DNA and RNA, there also the synthesis is blocked, and these antibacterial drugs have no ill effects on human because folic acid is not synthesized by the humans, but they are supplied through the diet. So as humans do not synthesize folic acid and they take it through diet, so they are not affected on the bacteria. They synthesize the folic acid and they are affected. Polysulfonamides acts as antibacterial agents. Methotrexate, you see the sulfonamides, antibacterial because of this inhibition of this folic acid synthesis and thereby DNA RNA synthesis, bacteria. Methotrexate and structural analog of folic acid again is used as anti-cancer drugs. Azacerin is a glutamine antagonist and blocks steps 2 and 5 reactions. And particularly 5, azacerin glutamine antagonist, they block the reaction 5. 6 markoptopurine inhibits the reaction 13 and 14, adversely affects the formation of AMP, GMP, and from IMP. So that 6 markoptopurine inhibits the reaction of 13 and 14, and thus adversely affects the formation of AMP and GMP from IMP. And that mycophenolic acid, a fungal compound, inhibits the enzyme IMP dehydrogenase, the step 14, and blocks the synthesis of GMP. Now, regulation of purine nucleotide biosynthesis. One is the de novo synthesis of purine nucleotides is finely coordinated to maintain the total pool of purine nucleotides available for nucleic acid synthesis and to maintain the proportionate concentration of ATP and JTP as per cellular needs. Purine nucleotides are interconverted to balance the levels of adenine and guanine nucleotides. And number 1, the concentration of PRPP. The concentration of PRPP is the primary determinant. So concentration of PRPP is the primary determinant of purine synthesis. The concentration of PRPP in turn is dependent on the availability of this ribose 5-phosphate and the enzyme PRPP synthetase. So concentration of PRPP is dependent on the availability of ribose 5-phosphate and the enzyme PRPP synthetase. Second, the enzyme PRPP glutamyl amidotransferase, and that is the the key reaction, and this the this is controlled by a feedback regulation. If AMP and GMP levels are sufficient to meet the cellular requirements, their synthesis is inhibited at step 2. So this is a reaction that, so if the AMP and GMP are sufficient, then this step 2, the reaction catalyzed by glutamyl aminotransferase, PRPB glutamyl aminotransferase is inhibited. And definitely through the same feedback inhibition, AMP and GM inhibits step 12. Through the same feedback mechanism, AMP also inhibits the step 12 and GMP inhibits step 14. And formation of AMP in GMP from IMP, so that is regulated. It should be noted that synthesis of AMP from IMP requires GTP as the energy source, while synthesis of GMP from IMP requires ATP as the energy source. And this reciprocity balances the synthesis of AMP and GMP. In other words, when there is adequate ATP in the cell, GMP will be synthesized from IMP, and when there is sufficient GTP in the cell, AMP will be synthesized from the IMP. Thank you, students. Today I am concluding with this class up to this. In next class, I will start with salvage pathway of purine synthesis.